So.
Buenos días a todos y a todas. Vamos a dar comienzo al tercer módulo del curso de microscopía aplicado al estudio de la respuesta inmune. Este tercer módulo está concentrado en la microscopía electrónica, que vamos a ver desde sus fundamentos hasta su aplicación. La primera charla va a estar a cargo de la doctora Graca Raposo. Good afternoon, Graca.
Good afternoon.
Good afternoon. It is a great pleasure for me. It is a great pleasure for me to introduce Dr. Graca Raposo. She's a group leader of the Structure and Membrane Compartment Laboratory at Curie Institute in Paris. At the beginning of her scientific career, after her PhD, she did a postdoc at the Immunology Center in Marseille, France, and then she moved to the Netherlands, where she had another postdoctoral position at Utrecht Medical School, and there she worked in B cells and MHC class II trafficking. Throughout this period, she was trained in electron microscopy, and in 2003, she returned to France and she established her team at Curie Institute. The major goals of her research are to gain a better understanding of the biogenesis and functions of two specialized endosomal organelles, which are exosomes and melanosomes. She has done fundamental contributions in this field, and her work pushed forward the knowledge of intracellular organelles biogenesis, and as she said once, the most satisfying discovery that she did was exosomes. She is highly recognized for her contributions in this area, which is reflected in the seminal and several publications that she has listed in her CV in different prestigious scientific journals. What is more, she's a reference in electron microscopy. Along her scientific career, she has received several distinctions from different institutions, such as NRS, Academy of Science in France, and the Society for Extracellular Vesicles as well. Since 2015, she is a member of the European Molecular Biology Organization, EMBO. I had the privilege to meet Graca during my postdoc at Curie Institute, and I learned a lot from her and her team. It was a great experience for me, and thank you, Graca, for that. I assure you that Graca is a very hardworking woman. She's very enthusiastic, not only in relation to research, but also academically. So, Graca, thank you very, very much for accepting our invitation to participate in this course. It is an honor for us. So now we are ready to listen and to learn from your presentation.
Thank you so much. I mean, first of all, I mean, before starting sharing my screen, really, gracias, Carolina. Gracias for everything. You know, for me, it was also a pleasure to work with you.
Welcome.
And I'm very, very happy to be here today with you. I mean, you in Argentina, me, I'm in France. And it's, I mean, this period, it has been very special, but we can continue communicating like that and sharing. And I mean, I'll be open to any question at the end or even after by email, okay? So I'm going to start sharing my screen. I'm going to start this seminar, you know, and it's a great pleasure to be here today with you and talk to you about one of my patients, that is electron microscopy. It's true that, I mean, I think that all techniques are important, but I hope to convince you that it's true that electron microscopy, I mean, looking on cells, looking on tissues, I mean, it's really, really important. So my title is The Past Makes the Future, because as you'll see, I mean, it is a very old technique that still have a great, great future. I'm really sure of that. So first of all, it's true that as Carolina said, I mean, one of my goals in my career, I've been really to study organelles, intercellular trafficking, understanding the biogenesis of these organelles, understanding the molecular basis of diseases. So it's examples that I'll give today, I mean, will be mostly on these intracellular organelles, showing several examples. But of course, the techniques that I'll present to you, that I'll try to share with you, may be applicable for other systems. So it's true that I'm sure that you have been convinced already yesterday from other talks that the importance of optical microscopy. But one thing is clear, you know, is that it's true that we have now great methods to do live cell analysis, even, I mean, with different fluorescent probes, and even to do a lot of quantitative analysis and image analysis. But I think that we have now this choice of complementary methods that go from the cellular level to the subcellular level. So today I'll be focusing more on the way of how we can localize components in cells, molecules in cells, and this will be more by the end of my talk. And in the first part of my talk, I'll go more to what we call these conventional electron microscopy, and some methods that can really bring more to our knowledge on intracellular organization and then organelle organization and biogenesis. So this is just to give an example. I'm sure that most of you have seen a microscope. This is one of the microscopes we have at Institut Curie. This is a Techni 120 kilovolt. I mean, this is a very user-friendly microscope, I would say. You know, of course, now they are equipped with digital cameras, and we are very lucky. Maybe Carolina, when she was in Paris, we were still developing these negatives, is maybe sure of that. Now we have this chance to have these digital cameras that are very appropriate to have directly the electron micrographs. But I always like to show this image of the first electron microscope that was built in 1931 by Ruska, Ernst Ruska, who actually shared the Nobel Prize in Physics later, much later after the war, because of course he had to leave during the Second World War, where he was able to continue his work during the Second World War. I mean, somehow, because he was born in Berlin, and he was somehow hiding to try to continue working on these electron microscopes. True that now sometimes you see in the publicities these great electron microscopes. I mean, and this is an example of these great Titan Cryos. This is a Cryo Microscope that is also made for other purposes. There are few around the world, but I mean, they are extremely expensive. They are very useful for some of the purposes that when you really need to do a very high resolution cryomicroscopy. But for the methods that I'm going to show you today, I think that a very basic microscope can be very sufficient. So this is just a principle that you have had already, but this is just to show you very basically how a microscope is built. First, you have a source of electrons with a high voltage. So you have a filament, which is either a filament of tungsten, or we use some cathodes that are long-lived because it's more easy to align the microscopes. And then you have the vacuum. So these electrons are accelerated in the vacuum. Then you have different lenses, and you have the electron beam, and the image is made on this fluorescent screen and camera. So basically, you know, in between, the image is just, I mean, it is just made by the diffraction of the electrons, which is that you have these electrons that are accelerated in the high vacuum. They go through the lenses, they hit the sample, and then these electrons, they are going to be deviated from the sample. So the image that you see is finally the contrast that is made, and you have here the example of the fingers and the head of the bull. So the image is clearly a 2D projection of the specimen, and as you see, of course, it's all black and white. And sometimes we say, oh, it's all black and white. Yes, it's all black and white, but as you'll see later, I mean, then we can put some pseudo colors, essentially, when you are doing electron tomography. So importantly, in principles, the specimens, as you know, they cannot be alive. So it's very nice to have these, the idea at the cellular level, using optical microscopy, you know, of the, basically the dynamics of organelles. Here we are fixed, the specimens are fixed, they are immobilized. Then we need to remove the water content of the samples. Then the samples are dried, so you have to keep these in heads. They are embedded in a resin that is resistant to the heating by the electrons. And of course, you know, we have to avoid damaging the sample. The thickness of the sample normally to do a regular analysis is in between 50 nanometers to 80 nanometers. Here I'll go to 400 nanometers as it's indicated in this slide, but this is a particular case that I'll show later. That is when we do electron tomography. And then, of course, you have this, the viewing of these 3D images from the specimen requires strategies, and I'll show examples of what we call this 3D reconstruction. So I'll go first through what we call this conventional EM, using normally chemical fixation. Chemical fixation is done normally with aldehydes, like glutaraldehyde. We can use a mixture of glutaraldehyde and paraformaldehyde. You know, we never use what we call formaldehyde. This is freshly made paraformaldehyde that you also use to fix samples for fluorescence microscopy. So we can use glutaraldehyde, paraformaldehyde, and as you'll see later, when we want to preserve antigenicity of the components, and this is what I show here as immunocytochemistry to localize molecules in the samples. So in this case, sometimes we use only paraformaldehyde because the antigens can be destroyed by the use of glutaraldehyde. They are not destroyed, they are just too fixed to be recognized by the antibodies. So this is just a patchwork of these different techniques. So you have the conventional EM here. I mean, somehow my arrow is not really working, so it's a little bit difficult. But you have the conventional EM, where you can appreciate here on the bottom, on the top, sorry, the Golgis, the different organelles, the mitochondria. In the center, you have an example of immunolocalization using two different types of gold particles with two different sizes. Again, you have a nice Golgi, you have an endosomal compartment. We can also use conventional EM to detect enzymatic activities, and this is a very old method, and I think I'll give you an example a little bit later. We can use enzymes that can then produce an electrodense product. Electron tomography, you see here an endosome that has been reconstructed like that. And today I won't go through, but I think it's also nice to keep in mind that there are now a lot of methods to do cryo microscopy. And cryo microscopy, when you are doing really very high resolution methods, and as you know, there was the Nobel Prize to Jacques de Bouchet about 2 years ago for the contributions he have made to cryo microscopy. So I like to show this image because this is a very old image. This is a pancreatic cell, and you have here all these reticulum, this endoplasmic reticulum. You see a bit of the nucleus, you see the mitochondria, and this is a whole old picture from over 50 years ago. And to tell you, basically the techniques haven't changed much. They haven't changed much since the beginning, and we are still using the same fixatives and using the same contrasting agents. I'd like just to make a note, because it's true that the agents that we use to contrast the samples, for example, uranyl acetate, osmium tetroxide. So it's true that now we are extremely careful in using these contrasting agents, because uranyl is derived from uranium. So of course we need radioactive waste, even if the radioactivity is very, very little. And so there is a lot of efforts to try to use now alternatives to this type of contrasting agents that are not really good for that are really, I mean, they are not very good for our health. So we have to be very careful when using it. I mean, there's a lot of toxic agents. Also, I mean, like the fixatives, of course, you have to be careful when using these fixatives. You have to use them under the hood, also using the resins. So we are all extremely careful when doing these techniques. You really need a chemical hood. This is just an example. As I told the organizers, I won't go very, very in detail into this, but you'll have the presentation if you want to keep it. This is just an example of what we do for processing for resin sectioning, for conventional microscopy. So you have samples, you have cells, for example, that you can put in a pellet, or you can even grow cells on cover slips like you do for immunofluorescence. Or you have pieces of tissues, then of course they need to be fixed, then they need to be contrasted. We generally use osmium tetroxide. Then, as I told you in the beginning, in the principles, they need to be dehydrated. We need to remove the water, and generally we use ethanol, increasing concentrations of ethanol before they are infiltrated in resins. There are different types of resins, epon. There are also mixtures of epon and araldite. And then we polymerize at 60 degrees for over 48 hours. Generally, you arrange to do it over weekend. So then once they are polymerized, so you see this takes about 2 days, I mean, including the weekends, to give you an idea of how long it is. It's not so long, finally. And then you just use a regular microtome. Here, the example I show is we use a diamond knife. So we have this diamond knife. You can see here on the bottom, the little sections coming into the water. So the sections of these resin embedded cells and tissues are cut. These are ultra-thin sections of about 60 to 80 nanometers. You can see a little bit the color. So these are interference colors that you can look on the loop of the microtome. And you have these sections, and then you collect the sections with an electron microscopy grid, these small grids that are around 3 millimeters. So normally we use a eyelash to manipulate the sections. You know, it's very homemade. You know, you have an eyelash that you put on the hood and stick just to manipulate this. Although everything is about to be automatic, I mean, you cannot just leave the microtome working alone, of course. So here it's a diamond knife. I mean, but for many years we have been using glass knives that can also be made, and in many labs around the world, I mean, I'm sure that glass knives are still pretty much used. The price of a diamond knife like that is about almost 3,000 euros. So, but you can keep it for long when you're careful with it. So this is an image that I pretty much like to show, because this is a dendritic cell with T cells. So this is a immunological synapse. So this was my my life before pigmentation in immunology, although I'm still very interested by, what immune cells have to tell us. And I like to show you because it's really to show you, you can see what's inside the dendritic cell. You can see these immune synapses with the T cells, and you can even measure, with antigen or without antigen. So, and this was just merely, embedding these cells that we have been together in the cover slips, and we could study the morphology of these cells and even the size of the immune synapses. So these type of methods are very, very useful to study intracellular compartmentation in health and disease. And when someone comes to see us and say, oh, okay, what can I do with it? Can we, I have even biopsies of a patient that have maybe metastasis of a melanoma into the liver. Now it's always difficult to, of course, when we are working close to the hospital. And of course, this is a method that we can do. We can take these biopsies and we can look, and we can use these very basic methods for many purposes. Here is just an example that I want to share with you, and this was done, work done in Institut Curie, for example, by Ilse Urbain in collaboration with the team of Sylvie Robin and Daniel Louvain some years ago. And you can see very nicely, just doing conventional EM, the microvilli. So this is the intestine of mice. There are knockout for some cytoskeletal proteins like aspin and fibrin. And you can even measure, on the microvilli here on the bottom, you see the microvilli that were cut on the other sides, because you can always orient your samples. So you can have your nice epithelium, or you can cut your villosities on the other side, and then you can see the organization of these of the interior of these microvilli. And you can do a lot of, it's always very important to keep in mind that, an image, an electron micrograph, but you can show one electron micrograph in a paper, but you always need to do some image analysis throughout, to show that there are really differences between a control and in this case, these mice that are knockout for these proteins. So, another example that I wanted to show is this case is using Cytos Chemistry. So this is from a paper that we had some years ago in collaboration with our colleague and friend, Mickey Marks. And here on the top, you see a control, and here we have used DOPA. DOPA is one of the precursors of the pigment melanin, and we just feed the cells with DOPA, that is a chemical, and we can see the activity of this enzyme, that is tyrosinase, that is a major enzyme that is synthesizing the pigments in melanocytes, in skin cells. And here you can see in the mutants how the enzyme is retained, in this case in the Golgi, because this on the bottom we have a melanocyte that is mutant for a very important complex that is important for a proper pigmentation of these melanocytes. But you can see very nicely, just using this conventional EM, after doing the cytochemistry, you can see the differences and see how the activity of this enzyme is retained here, in this case, in the Trans-Golgi network. So this is another example, also from a recent paper that we had in collaboration in the team with another team in Curie that is more in the translational department, and they asked us to look, for example, of breast cancer cells that were untreated or treated with agents that are used in chemotherapy like carboplatine. And here, I mean, just to give you some examples, you see the mitochondria and you see what these agents are doing to the mitochondria. So here, this was really an analysis of the morphology of these mitochondria in this case, and how this was related to the metabolism of these cancer cells. So you have on one hand the normal mitochondria on your left, and then in the middle you see that the effects that you have on these mitochondria by these agents. And again, we could do a statistical analysis that you can see in this paper that I've put here for reference. So it's true that until now, I mean, I've been using agents that are indeed aldehydes, and these aldehydes, one problem of these chemical agents is their penetration rate. So you have here on the top, you know, you have here proximal tubule cells. You have here on the top outer zone, and you see that the morphology is quite well preserved, but on the bottom, or even here on the middle zone, you see that the morphology is much less preserved. You see, I mean, that is not as nice, it doesn't seem as nice as it is on the top. And the problem is the penetration rate. And to give you an example, I mean, for example, a chemical fixation here, you have a specimen and you have a chemical fixation. The chemical will take 5 to 6 seconds to enter into the tissue, okay? So for example, if you take a C. elegans, when you are working with worms, for example, you take a C. elegans, you can put it in aldehyde, very concentrated, and I tell you that the C. elegans won't bother much. You know, you will continue swimming, you know, into the fixative. So for that, I mean, you have cryofixation that really started over 20 years now to be really developed. And this cryofixation really takes, I mean, the minimum takes milliseconds to do. So, and you have these physiological events that take 1 to 5 milliseconds, you know. So when you have events that are very, very, how do you say, very dynamic, I mean, for like, you know, in cells, like you have endosome dynamics, it's true that we are never sure that when you're using chemical fixation, we are fixing at the good time. So it's true that cryofixation is, when possible, a very good option. And it's true, this I always show, it's the high stage. So in this case, I'll give you a series of examples, because in this case, you preserve better the tissues. And again, as I'll show after, when we want to do, for example, 3D reconstruction, I mean, you would like to start on a good base. And for that, it's true that, I mean, when it's not possible, you can do electron tomography on aldehyde fixed samples. Although the best is to really do this cryofixation, because then you can be more sure that you are close to native state. You preserve fusion-fission events when we are studying contacts between organelles. You can preserve fragile organelles. And it's true that the morphology is more near to the physiological state. One thing is that you preserve also the volume of the organelles. And I think, I mean, here I'm giving you an example. I mean, this is one of my favorite cells. So these are skin cells called melanocytes. They have these organelles. I won't enter really into detail, but you can see here different examples of these pigment organelles where, I mean, you have the pigment melanin in dark, but you can see here in the center these organelles with these small fibrillar structures. You can see one that is very close to the endoplasmic reticulum, and you see these fibrillar structures, they have been optimally fixed using this high pressure freezing. So And the volume of the organelles is also maintained, because the aldehydes, what they do is that, and because you dehydrate also with ethanol, is that you have shrinkage of the organelle. So here we maintain the volume, and we maintain the volume of the Golgi stacks, for example. It's very nicely preserved, I mean, for the cytoskeleton and all the vesicular components. I think I have here just an example from a paper in 2001. Here it's chemical fixation. I mean, maybe you'll say, okay, it's also nice. But if we are going to measure the organelles, I mean, you can really see a difference. Although we can also see these very nice little fibrils in these immature melanosomes that don't have melanin yet, they are much less preserved. And with our experience, I mean, we know now that using high pressure freezing, I mean, we can preserve much, much better all these structures and all these contacts. So here, this is again an example. You can see here very, very close contact. Can we detect maybe a fusion event between? But in also, and I'll come to that later to show you how we can do it. But you can see here also very nicely, for example, microtubules and all these vesicles around with the optimally preserved volume. So this is how it is. I mean, this is one example of one of the equipments that is commercially available. Maybe later in the afternoon, Xavier Heiligenstein will show you the equipments that they are now developing that are extremely interesting also. This one here is from Leica. So the cells can be cultured on sapphire discs, very small discs, because I mean, you cannot use glass, but you have other types of tubes and carriers that can be used for tissues, for example, or to carry, for example, little pieces of samples. And the rapid freezing, this is what we call this high pressure freezing. It really reduces the rate of ice crystal nucleation. You know, it reduces the, I mean, it's, you really need to, it has to be quick. It's quick by the cold and the pressure. This is what you have to, the take-up message. And importantly, cells in tissues up to 200 micron C can be frozen, you know, without vitrification. And this is from a paper from Martin Sachs and Ilse Urbain, great engineer in our lab. We've been really developing also this technique and doing also a lot of electron tomography. And they have worked a lot on the way that how you can really reduce and preserve samples to do this high pressure freezing. I show you here this because, I mean, sometimes, I mean, this is what happens. It's not easy to avoid ice crystals. And this cell that I'm showing here was just close to the other cell that was very nicely preserved. So this is really ice crystals. And sometimes you say, okay, I can recognize Golgi, I can recognize mitochondria, but here you have ice crystals. And it's very difficult to really avoid that. Again, I mean, this is another example of another equipment that was also available some years ago. This is one of the first ones that was developed by Leica. And how do you do it though? So after, because the specimens are frozen, then you have to do a process that is called freeze substitution. Again, this is an example of a protocol. Here, I mean, we can use sections of about 60 nanometers, like, I mean, for doing the conventional EM. And just to do a comparison between what we call this conventional EM with the fixatives, with the aldehydes and the high pressure freezing, it's true that you'll see that high pressure freezing, I mean, you don't do it routinely, but it's the optimal method when you want really to go further into the morphology of your tissue. And you can see here the differences. I mean, it takes more time. Of course, it's more expensive because you need another equipment. But I mean, easily, I mean, we can have access to these equipments. I mean, sometimes we don't have it in the lab, but we go to other labs, and these type of equipments are generally shared in between labs. So is this information enough? As I told you, I give you again here the example. When I can see here my melanosome close to a vesicle, is this a fusion event? Is this merely a contact? So here we need what is called electron tomography to be able to do it in 3D. So again, I mean, this is a pomegranate. I always like to show this pomegranate because you see the slices, but really to see what's in the middle, you need to use this kind of techniques. And for that, you have electron tomography where you have your sample. And then you have now, you need, of course, seeker samples, and you have softwares in the electron microscopes that will be able you to record the different images by tilting the specimen. You have your specimen holder in the microscope, and then the specimen is going to be tilted, and each time you have these images that are taken. So again, you have to take seeker sections, 200 to 350 nanometers. Then you put it on grids, and then you have to use what we call these markers, fiducial markers. Sometimes we put gold, just to be able to align the samples when you go into the microscope. So this is an example of an automatic image acquisition. You can leave your sample. Now the microscopes are equipped for that. And then you have a lot of treatment, of course, after that. You have, this is one of the first examples used to study multivesicular endosomes. These are multivesicular endosomes in B lymphocytes. Again, these are one of my favorite. This was from the lab where I did my postdoc in Utrecht. And this was extremely important to show that these vesicles that you have in multivesicular endosomes are vesicles and not merely tubules that are still connected to the outer membrane of the endosome. And I've put you here the reference because this is an AS paper. This is an example from our lab. So you have the acquisition, the alignment, the reconstruction, the segmentation, and these are two types of melanosomes in skin cells. And I mean, I gave you here an example of a reference from Ilse Urbain. I mean, this is in 2008, I mean, but we are in the COVID still period, I mean, all around the world. And I wanted to give you an example also of a paper that was made during SARS-1, by the way, but how they have used already electron tomography in viral cells to study, I mean, how the importance of the endoplasmic reticulum in these cells that were infected by the coronavirus. And last but not least, I mean, other methods of tomography for whole cells that you can maybe hear is, for example, focused ion beam or 3D view, because the electron tomography that I've given you the example, you can go to study an organelle, to study two organelles, but again, to have the whole view, maybe here you have less resolution on a particular organelle, but if you want to have the whole cell, you have now these methods called FIB, focused ion beam, or 3D view. So do I still have time, Carolina?
Yes, you have 5 minutes more if you want.
Okay, so it's just to give you now an example now of the how to localize, how we can localize components in macromolecules and tissues. So this is what I call you correlation, ultrastructure, and composition. So this is called immunocytochemistry. So you can use direct and indirect methods like you do for immunofluorescence, using, I mean, different antibodies, monoclonal, polyclonal antibodies. But in the case of EM, of course, you have to use electron dense products. I mean, the example here is, for example, horseradish peroxidase, but generally what we use is these colloidal gold particles that we can have in different sizes to do single, double, or even triple labelings. And there are different objectives. Of course, we have to avoid extraction, denaturation, steric hindrance, alteration by chemical fixatives. And you need high affinity antibodies, good accessibility of antibodies. You need to preserve antigenicity, as I told you in the beginning. Sometimes using glutaraldehyde is too hard. I mean, you cannot preserve antigenicity sometimes for some antibodies, but at the same time, you need to preserve ultrastructure. So you have different techniques, pre-embedding, labeling, then sectioning, but I'll give you just an example of post-embedding, sectioning, and then you label. And these examples, I mean, there are two examples, resins and free substitution, but this is quite particular, and I'll give you today more the examples of the technique of Tokayashu that we have been using for many, many years. And Tokayashu, I mean, died some years ago, and he really developed this technique very, very early on. So this is an ultramicrotome with a liquid nitrogen container. This is one of the most used, but the other ones are still working very nice. And here you cut with a diamond knife, but the diamond knife without water. So I just show you here a little movie just to show you how the sections then are made, because they are not collected on water. Again, you can use the high lash to do the section. You see the sections. You just have to be very patient and to be relaxed to take your sections, and then you're going to collect your sections with a loop. And normally in this loop, you're going to collect the sections. As you see also here, they are a little bit goldish, and these sections are also about 60 to 80 nanometers. It's by the color that we can really appreciate the thickness of the sectioning. So then is going to come with this loop up. And he collects the sections, and we put the sections on the grids. So this part is really, really tricky, because you go from what is very cold to the ice to 4 degrees. So this is just a protocol that Daniel can keep in the presentation, how to do the immunolabeling. I mean, there is a lot of advantages using this technique, because it's relatively quick and results can be obtained in 2 to 3 days. And of course, you can do quantification, you go to the microscope. This was some years ago. I mean, some years ago, it was very difficult, but there's a lot of improvements that were made over these years, essentially by the lab in Utrecht. And now you can really preserve very, very well the ultrastructure together with the immunolabeling. Again, these are some of my favorite cells, these melanocytes with the melanosomes. We have here 2 examples, big gold to label endosomes with the early endosomal antigen 1, and the melanosomal protein with the smaller gold. And here you have, of course, the picking up of the sections is one of the tricky things, but there's a way to bypass it. Again, you need to know very well the biology of your antigen. Always use negative controls. Again, I cannot insist more in the fact that we need statistics, we need to do image analysis, and because we can not only show the example of 1 micrograph in a paper. I mean, the reviewers always ask for statistics, and this is just one example from one of our papers. But as you'll see in most papers using these techniques, I mean, there are methods and a lot of methods to do the quantification, I mean, of the gold, even under the microscope or now, just on the computers, you take different images and you just count. So again, I mean, this is just the end, and to introduce the others, of course, as you'll see, we need all of these complementary techniques to analyze I mean, the biogenesis, to analyze intracellular localization. And so combining all these methods, and as you'll see later with Claudio and Xavier, I mean, there will be a lot of developments that are made now to really do these correlative light electron microscopy methods. And to the end, of course, it is a marvelous technique, electron microscopy. You know, you just have to be aware that sometimes of the artifacts, as I told you, when we use chemical fixation, we know that we have limitations, we know that we have artifacts, and of course, you can always be eaten by the crocodile. So when we have a positive result, I mean, we are always careful in trying to understand if this is real, if the antibody works. I mean, you really need to know the biology of your antigen and of your antibodies. And of course, last but not least, I mean, all the people in the lab, and I mean, my great colleagues, indeed, Ilse Urbain and Maryse Romao, the two engineers that really have been training a lot of people in the lab, and my colleague Claude Tony, that is now emeritus in the lab, that together we have set the lab when I arrived in Paris after my postdoc in the Netherlands. And thank you all for everything. On the other side of...
Thank you, Graca.
Yes, I'm sorry that it was a bit long, but...
No, it's okay. You were very clear, and the presentation was really interesting. We have some questions, and yes. One which is a more general question is, what consideration should we have before thinking about performing EM? I think regarding samples, experimental procedures.
I think for me, what is really important is, okay, I mean, you have your cells, you have your biological question, isn't it? This is the sense of your question. You know, you have your biological sample. You know, you are trying to understand, for example, if your cells, you are studying cells from a patient, for example. I mean, the example that I'll give you is, for example, immunological disorder. Okay, for, let's say, a syndrome of immunodeficiency. You know, for example, you have cytotoxic T cells. I mean, because I'm talking more for the Immunological Society, I'll take more this example because this happened to me some years ago. Is that we had cytotoxic T cells from patients, and these cytotoxic T cells, they are very small, so it's very difficult to analyze by immunofluorescence. And we just wanted to see what was the defect on the cytolytic granules, okay? So we just took normal cytotoxic T cells, you know, isolated from a normal donor. And then we took the cytotoxic T cells from the patient. And then we say, I mean, what is the first technique that we're going to use? Okay, the first technique that we're going to use, we are going to use conventional electron microscopy. You know, just fix the cells, embed the cells in resins, like I showed you in the beginning, use, you know, a protocol that can be adapted, that is adapted to these cells that are in suspension in this case. And then we're going to look what the cells have in their belly. You know, let's look on the morphology of these cytolytic granules in these patients in relation to the control. So normally I'll go to just do conventional electron microscopy. And then, you know, if we really see something that we say, okay, but now it will be interesting to really localize some of the components, because in the meantime, maybe the genes that are have been sequenced, we know the components that have been that are mutated in these in this disease. So now we are going to take the control cells, and we're going to see where the gene is located in the cytotoxic T cells. So we'll go by phases. For me, I'll just recommend to do conventional EM, not go directly to localization. And then, of course, when you are doing the localization of your components, you are going to first to say, okay, do I have antibodies? Are my antibodies recognizing working for EM? Because one thing that you know that is very important is that sometimes a lot of antibodies work for immunofluorescence, but it's not because they work for immunofluorescence that they are going to work for EM, because you are in different conditions, and you have always to take into consideration that by fluorescence you are looking to an ensemble of molecules, and on EM, because we are on sections, we have much less antigens that are exposed. And we always say 1 gold particle, when I show you 1 gold particle corresponds to 100 molecules, you know, still. So sometimes for some proteins, it's extremely difficult to really localize these proteins because they are not abundant. And this is very difficult.
Yes, it's true. Another question says, it's possible to estimate the volume of the organelles, for example, the mitochondria by TEM?
I mean, yes, I mean, it's possible to estimate the volume. I mean, of course, doing, the optimal volume, you can only have it using, of course, high pressure freezing, okay? Then you can do 3D reconstruction. Okay, you can do the 3D reconstruction to estimate the volume. Or, I mean, in the past, what people did was our serial sections. This is still possible. You know, you can take, you can see, for example, you take the cells and then you do this serial sectioning, and you go throughout the organelle, and you can then estimate the volume. But there are a lot of techniques. You know, there is this black book. I don't know if you remember when we were in the EM courses, there is the magic black book of stereology in electron microscopy. So I would recommend this magic black book that is extremely old, that a lot of people forgot. I should have put also an example. There is this the green book of Gareth Griffiths. Maybe I can send you later, because this green book of Gareth Griffiths is still, you know, for us a reference in the field, I mean, of stereology in EM. But of course, it's possible, yes.
Yes. And the other one says, do you have any recommendations for the immunostaining of cytosolic proteins which are not bind to vesicles? I mean, because in some cases this protein can be lost during the procedure.
Of course. I mean, and you touched there, I mean, an important point that I went quite quick. Because it's true that when we have proteins and components that are inserted into membranes, transmembrane proteins, you know, when we're using transmembrane proteins, of course, we fix the samples, we cut the samples. Then we can keep the grids. I didn't insist much on this part of the protocol, but we can keep the grids after the picking up for around a week in the fridge. But for cytosolic proteins or proteins that are just linked to the membranes, for example, with the GPI, for example, the prime protein. These proteins, although the cytosolic proteins and the proteins that are not transmembrane, I mean, what I can recommend is that after fixation, you label your samples immediately. You can even label on ice always without letting, you can increase the time of binding of the antibodies, because in the cold is a little bit, it takes more time. But you have to minimize the extraction of these antigens that are cytosolic, or even inside the nucleus. It's now very interesting to really understand the subcompartmentation within the nucleus, because you have the nucleoli, and I'm sure that we have compartments in the nucleus. And these proteins, again, I mean, even if they are fixed, they are going to continue moving because they are not attached to membranes. So the only thing is to label, cut, and label. And try to fix with glutaraldehyde if the antibodies work on glutaraldehyde. One important thing, I think that when we don't know an antibody and an antigen, of course, we have to be sure that by immunoblot you have one single band. It's extremely important not to start buying antibodies that are commercially available without testing them, because a lot of these antibodies, sometimes they recognize different proteins. There were a couple of examples like that. So you really need to know your antibody. And then you can fix your samples when you don't know. You fix your samples with performaldehyde and with a mixture of performaldehyde, glutaraldehyde. And then you try both in parallel. Just with a single label, increasing dilutions. You use dilutions. Normally, if the antibodies are already have been already used for immunofluorescence, generally we increase the concentration by 10. If you dilute an antibody 1 in 2,000 for immunofluorescence, I will try dilutions at 1/200. But again, the time of the affinity of the antibodies, if they are good after 15 minutes, the antigens have been recognized by the antibody. So generally we incubate for about 30 minutes. But when you don't know, just fix with 2 different fixatives. And it's, did I answer to your question?
Yes. Okay. Yes, thanks. And the last one, because we have to move to the next speaker, is do you have any experience in immunolabeling, sorry, of bacteria?
Of bacteria?
Yes.
Yes, I mean, myself, I've not done that, but of course, I mean, bacteria inside cells, yes, I mean, there's a lot. And I would recommend, for example, looking on the papers from Peter Peters from the Netherlands. Remember Peter Peters? Yes, he worked a lot on mycobacterium. And of course, I mean, bacteria, you can embed the bacteria also in gelatin, as we do normally for the cells. You can embed the bacteria, and then you can do sections, and you can immunolabel, you know, in your pellets. And but I would recommend, yes, looking at I mean, there's a lot of methods, you know, now to do immunolabeling on worms, on Drosophila. We have been working lastly more on Drosophila. It's, I mean, it's tricky. It's tricky, you know, because then you need to embed only, you know, one part of these model organisms. I think it's the part of the model organisms is very challenging, and I can also recommend looking, for example, a paper from Guillaume Vanille in a dev cell, for example, where they have cut pieces of zebrafish and do immunolabeling on these pieces of zebrafish, for example.
Okay, thank you very much.
I mean, if you want to email, just email questions, and if I can help, I'll be happy to do it.
Okay, thank you, and enjoy your holidays.
Yeah, not yet. But what I'm gonna do is, so I think I'll stay a little bit for the other for the talks of Claudio and Xavier.
Okay.
I'll stay a little bit with you. Okay.
Okay. Thanks.
Bueno, vamos a continuar con la siguiente charla. Es un placer para mí presentarles al doctor Claudio Bussi. Claudio Bussi ha sido estudiante de bioquímica en la Facultad de Ciencias Químicas en la Universidad Nacional de Córdoba. Muy entusiasta ya desde estudiante, obtuvo 2 becas SIN. Tuvimos la suerte de que se una a nuestro laboratorio. Se recibió en 2012 de bioquímico y obtuvo la medalla de oro al mejor promedio. A partir de ahí se sumó y realizó el doctorado en nuestro laboratorio, interesándose por la neuroinflamación y particularmente por la autofagia, aplicando numerosas metodologías. Y particularmente le interesó mucho siempre el estudio de las imágenes, y tuvo la posibilidad de hacer pasantías importantes en Cambridge y en IMBL en Alemania, en la cual pudo hacer estudios de los autofagosomas aplicando CLEM y LibCLEM. Actualmente Claudio se encuentra haciendo un postdoctorado en el Instituto Francis Crick Institute de Londres, y su excelencia en el manejo de las imágenes y en el procesamiento de la microscopía se pudo ver reflejada en una publicación en la cual en la revista Journal of Cell Science le dio, colocó una de sus imágenes, una de su trabajo artístico en la tapa de la revista. Así que hoy nos va a hablar de principios de aplicaciones de microscopía correlativa, CLEM. Así que bueno, cuando quieras, Claudio, bienvenido. Te escuchamos.
Hola, bueno, muchas gracias Pablo por la introducción. La verdad me siento un poco intimidado con el resto de los panelistas. Las charlas anteriores estuvieron muy bien, así que espero que puedan disfrutar esta. Empezar acá a compartir la pantalla. Bueno, antes que nada quería agradecer y contarles también un poco mi experiencia, cómo llegué al CLEM, que me parece que puede ser de utilidad también para los becarios y becarias que quizás quieran introducirse en esta metodología. Esto empezó con un curso que yo hice en el IEM, en Mendoza, como parte del Sistema Nacional de Microscopía se dicta este curso, y aquí también había gente del MBL, del Laboratorio Europeo de Biología Molecular, que ellos son bastante avanzados en el desarrollo de esta metodología. Y a su vez el grupo de la doctora Colombo en Mendoza es uno de los pioneros en el uso de CLEM en Argentina. Y entonces, bueno, fue una experiencia muy buena, y ahí es donde empezó todo, digamos. Después quiero agradecer también al Laboratorio Europeo de Biología Molecular, que me alojó 4 meses, y en particular al grupo de Yannick Schwab y a la Facility de Microscopía Electrónica, y luego, por supuesto, a la FSQ y a la UNC, que es donde pasé gran parte de mi vida, diría. haciendo el doctorado, bueno, en la carrera de grado, así que bueno, un saludo grande a todos los de allá, espero verlos pronto, en especial abrazo al CBC. Y en particular, bueno, quería agradecer al laboratorio de Pablo y al de la doctora Selej, porque fue donde, ya van a ver más adelante, donde yo voy a introducir la proteína con la que yo estuve trabajando durante mi doctorado. Fue una colaboración que surgió entre nosotros y el grupo de Sole Selej y la ayuda de José Ignacio Galea. Bueno, ustedes ya tuvieron una charla espectacular de la doctora Graca Raposo, donde ella introdujo de una manera muy clara todo lo referido a la parte tecnológica de la microscopía electrónica, y yo lo que vengo a mostrar acá son básicamente ejemplos y aplicaciones de CLEM, que básicamente, en otras palabras, es microscopía de luz y electrónica en la misma muestra. Es simplemente eso, pero obviamente tiene distintas complejidades dependiendo de qué es lo que queremos responder. Y esto es, para mí, lo más importante que tenemos que tener en cuenta es la pregunta, ¿sí? ¿Qué es lo que estamos estudiando? ¿Nos hace falta CLEM para responder eso o no? ¿Qué otras metodologías puedo usar que quizás me ayuden a llegar a ese punto de una forma más aproximada, o realmente necesito usar esta técnica? Entonces, acá yo enumeré algunas preguntas. Yo creo que es una metodología fabulosa y que nos va a permitir descubrir un montón de mecanismos que no conocemos. Y, por ejemplo, si uno quiere saber cuál es la localización subcelular de la proteína de interés que tengamos, cualquier proteína, ¿sabemos realmente cuál es la localización subcelular? O, por ejemplo, la señal fluorescente en la que nosotros confiamos, ¿podemos decir en el 100% de los casos que corresponde a una estructura particular determinada? Entonces, si la respuesta es no, o quizás no, es una buena manera de introducirse en esta metodología. También, si queremos investigar cómo la localización puede tener un impacto en la función, si la estructura 3D es conocida o no, y si esa estructura 3D puede a su vez resultar en una funcionalidad particular o no. Y luego, algo que también en la charla anterior se discutió un poco, y yo creo que va a ser probablemente uno de los campos de más desarrollo, es todo lo relacionado con la biogénesis de organelas y la regulación. Entonces, yo acá les voy a mostrar brevemente lo que sería el protocolo para realizar CLEM. Este es el que yo usé en mi estadía en el MBL. Básicamente va a comenzar con un cultivo celular. Esa parte del cultivo celular probablemente vaya a ser expandida en la charla que sigue, porque hoy hay diversas estrategias en cómo vamos a cultivar esa célula dependiendo de qué metodología de microscopía electrónica se va a usar después. En mi caso, lo que yo usé fueron discos de zafiro, y en estos discos se adhieren las células. Esos disquitos están en una placa de cultivo, ahí se van a adherir las células, nosotros vamos a hacer el tratamiento X que querramos realizar, y luego eso va a ser fijado. Que en el caso de mi experiencia, yo usé la técnica de congelamiento o congelación en alta presión, que es HPF por las siglas en inglés, que nos permite una óptima conservación de las membranas, como ya se discutió en la charla anterior, que es una gran ventaja en relación a la fijación química, que sería la más común. Pero bueno, también, como se discutió anteriormente, esta técnica presenta la desventaja de que necesitamos más tiempo para desarrollarla, y también es más costosa. Por lo tanto, no es la rutina de todos los laboratorios. Luego de la fijación, uno va a realizar este proceso que se llama freeze substitution, que básicamente es el comienzo de lo que va a ser nuestro armado del bloque de resina. Esto va a llevar como una semana aproximadamente y puede ser realizado de forma automática por robots que realizan este proceso. O sea, uno nada más agrega los distintos reactivos y después el robot va mezclando, y luego de una semana tenemos nuestro bloque de resina. Y luego, una vez que tenemos el bloque, que obviamente contiene nuestra muestra, va a ser seccionado, y luego lo que vamos a necesitar hacer luego de esta sección es este paso, que es opcional, pero muy recomendado cuando uno quiere tener precisión en lo que queremos localizar, que es la agregación de fiducials, o acá le dicen esferas fluorescentes, que son electrodensos, son partículas electrodensas, muy chiquitas, pero también fluorescentes. Y esto nos va a permitir después hacer un alineamiento muy preciso una vez que tengamos la imagen final. Ya todo esto va a quedar más claro cuando comience a mostrar algunos ejemplos. Luego de este paso, que hasta ahora lo único que tenemos es un corte, se hace este ensamblado en sándwich, que les voy a mostrar en la próxima slide, y a ese corte, que está ahora ensamblado, lo vamos a llevar al microscopio, ya sea confocal, de super resolución o wide field. Vamos a adquirir las imágenes, y posteriormente esa sección va a ser colocada en una grid de microscopía electrónica, y luego se va a realizar la adquisición en el microscopio electrónico que hayamos elegido. En mi caso lo que hicimos fue tomografías en secciones de aproximadamente 200 o 300 nanómetros. Luego se realiza una reconstrucción de esos tomogramas, y finalmente el proceso de correlación, que eso se va a realizar mediante software, donde uno de los software más utilizados es el EasyClem, que seguramente Javier puede comentar en la charla siguiente, porque él ha sido uno de los principales contribuyentes a ese software. Aquí esta es otra review que yo les dejo para los que quieran interesarse en otras estrategias. Acá básicamente lo que está haciendo este gráfico es hacer un resumen de lo mismo que yo ya comenté anteriormente, pero mostrando también la otra opción, que es todavía, yo diría, un poco más avanzada y solamente necesaria si uno quiere visualizar estructuras de proteínas en relación con membrana. O sea, si ya queremos ir al detalle casi nativo de proteína, se hace básicamente todo en Cryo, y si no, es digamos, el protocolo más común es este que acá sale como RT por temperatura ambiente CLEM. Entonces ahora, para que quede un poco más claro con distintos ejemplos, yo voy a comenzar con mi experiencia durante el doctorado y luego voy a mostrar algunos otros ejemplos de publicaciones. Entonces, como discutimos en la primera slide, o la segunda, en relación a cuál es la pregunta que queremos resolver, durante el doctorado yo estuve estudiando el proceso de autofagia, y el proceso de autofagia posee una organela clave, que es el autofagosoma, acá la pueden ver en el gráfico más a la izquierda, que es una estructura de doble membrana, y que se encuentra decorada por esta proteína que se llama LC3. Pero luego, hará unos 10 años aproximadamente, un poco menos, en realidad si 10 años, un poco más, 2008, se descubre este otro proceso, donde LC3 se asocia a estructuras de única membrana, como un fagosoma. Y entonces esto empezó a cambiar y abrió, en definitiva, un nuevo campo de investigación. Y esto en realidad también fue posible gracias a la microscopía electrónica. O sea, fíjense cómo una observación, en definitiva, ahora resultó en un nuevo campo de investigación, porque uno antes pensaba de que el LC3 iba a estar siempre en estructuras de doble membrana, y se demuestra de que el S3 puede también marcar estructuras de simple membrana, como el fagosoma, y este proceso se llama fagocitosis asociada a LS3. Entonces, yo estaba estudiando este proceso luego de la internalización de la proteína alfa-sinucleína, y quería responder si el LS3 estaba marcando, en mi sistema estaba marcando una estructura de doble o de simple membrana. Entonces, obviamente, esto no me lo iba a permitir resolver la microscopía de fluorescencia, y para ello fue que era necesario realizar CLEM. Acá, en la primera figura, lo que les muestro es el armado del sándwich. Acá ya se ve la grid que va a terminar siendo usada para microscopía electrónica, que contiene a nuestra sección con la muestra. Entonces se arma este disquito que se va a llevar al microscopio de fluorescencia. Vamos a adquirir las imágenes como las que yo les estoy mostrando acá. En mi caso yo estaba interesado, como comenté, en la proteína LS3. Esto fue desarrollado en células microgliales BB2, murinas, que expresaban establemente LS3. Alfa-sinucleína era mi proteína de interés y la teníamos marcada. Y en este caso estoy mostrando una tinción con lysotracker porque también habíamos observado de que principalmente alfa-sinucleína colocalizaba con vesículas lysotracker positivas. Luego, a estas mismas células, como pueden ver, la vamos a poder observar por microscopía electrónica, y el resultado final, esto es un primer merge o overlay que se hace, nos va a permitir decir, bueno, ¿qué zonas ahora queremos adquirir? En el final de la charla anterior se comentó de que se puede realizar una adquisición de imágenes automáticas, y esto la verdad fue increíble en cuanto a la cantidad de datos que uno puede generar, porque básicamente uno puede seleccionar todos los puntos o áreas de interés que nosotros tengamos o estemos interesados en resolver, y el microscopio va a ir uno por uno adquiriendo esas áreas y haciendo tomogramas de cada uno de ellos. Entonces, luego el resultado final es algo como lo que les estoy mostrando en el panel inferior, donde tenemos la microscopía de fluorescencia, la microscopía electrónica, y luego finalmente la magnificación del tomograma ya luego de que se hizo la alineación y demás por microscopía electrónica. Y entonces acá lo que pudimos observar es que LS3 estaba asociado a vesículas de doble membrana, o sea, a autofagosomas, y no vesículas de simple membrana. Ya las voy a mostrar en más detalle en la próxima diapositiva. Pero también, como pudimos analizar cientos de estas áreas, encontramos que el LS3 no necesariamente iba a estar siempre en estas vesículas de doble membrana. Yo creo que eso es una de las principales ventajas y curiosidades, si se quiere, que vamos a encontrar con la microscopía electrónica y con CLEM, si podemos analizar muchas áreas de interés. Y eso realmente despierta la curiosidad y potencialmente permite el desarrollo de nuevas áreas, porque es realmente sorprendente ver en la diversidad de estructuras que vamos a encontrar a aquella proteína que uno originalmente pensaba que pertenecía a un solo compartimento. Acá les voy a mostrar el mismo ejemplo de la figura anterior, donde a la derecha se va a apreciar la formación de la vesícula del autofagosoma, de la vesícula de doble membrana. Fíjense cómo podemos ver la primer bicapa y luego la otra bicapa de fosfolípidos, y la estructura que está a la izquierda, la estructura más grande, es de simple membrana, y por la tinción que nosotros hicimos, podemos suponer de que es un lisosoma o un autolisosoma. Y fíjense cómo también se puede ver en detalle las distintas vesículas que a su vez lo componen a este lisosoma. Este es otro ejemplo, donde aquí el LC3 fue asociado principalmente a esta estructura de simple membrana. Si uno presta atención, la membrana que está delimitando toda el área exterior es simple, pero a su vez también podemos ver cómo este compartimento está lleno de otras vesículas. También fíjense qué bien que se preservan las membranas, porque estamos usando esta técnica de congelación en alta presión, y este detalle no lo vamos a lograr si usamos, por ejemplo, paraformaldehído con glutaraldehído. Pero, ¿qué pasa? También podemos empezar a adicionarle cierta complejidad. Esto en realidad surgió luego de estar discutiendo con los chicos allá en el MBL, que me dicen, bueno, pero al final en una de esas esto es algo aleatorio. Uno obtuvo ese evento donde hay un autofagosoma, colocalizó o correlacionó con el S3, pero no necesariamente pasa siempre eso en el proceso que yo estoy estudiando. Entonces, lo ideal en este caso era realizar CLEM, pero luego de Live Cell Imaging. Y eso es lo que yo les voy a mostrar ahora. Entonces, acá de nuevo tenemos a estas células BB2 que expresan LC3 en verde y que internalizan a las fibras de alfa-sinucleína. Acá en el video se puede ver cómo el LC3 va a polarizar hacia un área en donde están estas fibras y forma esta estructura de tipo anular. Y acá lo pueden ver también en distintos fragmentos más pequeños a lo largo del tiempo, cómo se va formando esta estructura de tipo anular alrededor de las fibras de alfa-sinucleína. Entonces, aquí luego lo que hicimos fue fijar, en este caso, por cuestiones de tiempo, tuvimos que usar fijación química, o sea, paraformaldehído o glutaraldehído, pero aún así pudimos encontrar que esta área en donde ustedes pueden ver que tenemos la señal para ambos marcadores, correlaciona con una vesícula de doble membrana, o sea, con un autofagosoma que lo pudimos reconstruir en 3D. Y ahora les muestro este resultado en la próxima diapositiva. Entonces acá lo que se realizó fue cortes seriales de esa área, y cada corte fue luego utilizado para una adquisición de tomograma. Entonces acá lo que se hizo fueron aproximadamente entre 15 a 20 cortes de aproximadamente 300 nanómetros cada uno, y luego se adquirieron tomogramas de cada uno de estos cortes. Luego esos tomogramas son alineados y nos permiten a nosotros tener una reconstrucción en 3D de al menos ese volumen adquirido. Lo importante acá para mí también para recalcar, y que esto fue todo nuevo porque yo estaba recién introduciéndome en esta técnica, es la ventaja de poder hacer adquisiciones seriales, porque si uno quizás solamente tenía una de las otras secciones, se perdía el detalle donde íbamos a encontrar a la vesícula de doble membrana. Por ejemplo, acá en la imagen de la derecha, yo solamente es una de las varias slides de las que están en la película esta de acá de la izquierda, pero podemos ver cómo se aprecia una doble membrana. Sin embargo, si uno ve la película esta desde el inicio, ve que no siempre fue clara esa estructura. Entonces por ahí uno se pierde algún detalle por no tener la oportunidad de hacer 3D. Este es otro ejemplo de otra célula que fue también, que también hicimos Live Cell Imaging. Pudimos encontrar esta estructura de multimembrana que contenía en su interior a una de doble membrana. Esto va a quedar más claro ahora en esta reconstrucción 3D. Se pueden ver todas estas estructuras de multimembrana y la estructura de doble membrana al interior. Y luego pudimos reconstruir, y lo que está en distintos colores son membranas que por la característica que tiene serían membranas del retículo endoplásmico. Y esto la verdad también nos alegró en cierta forma porque reflejó bastante uno de los trabajos pioneros en tomografía y reconstrucción 3D de autofagosomas, donde se ven a las membranas del retículo endoplásmico rodeando al autofagosoma que estarían naciendo a partir de estas membranas. Bueno, ahora lo que hice una selección de otros ejemplos para que puedan imaginar potenciales aplicaciones. En este caso, este es un trabajo del grupo de Facundo Batista, donde lo que ellos observaron era ellos observaron en células B que estaban estimuladas con un antígeno, en este caso anti-inmunoglobulina M. Luego de la internalización del receptor de células B, encontraban este patrón en forma de anillo cuando hacían microscopía de superresolución, en este caso microscopía SIM, como se puede observar en el primer panel. Y si uno hace el overlay con la microscopía electrónica, ellos observaron que la mayoría de estas estructuras de tipo anillo que contenían al antígeno eran principalmente estructuras de simple membrana. O sea, acá la pregunta era en cierta forma similar a la que yo también estaba intentando responder con mi trabajo, para ver si quizás la autofagia canónica o no canónica, una de las formas de la no canónica es este proceso asociado a fagocitosis, donde LC3 estaría solamente marcando estructuras de simple membrana. Entonces, bueno, querían responder un poco eso junto con, por supuesto, toda la otra evidencia de biología molecular que ellos ya venían estudiando en este trabajo. Pero encuentran de que la mayoría de estos anillos son vesículas de simple membrana. Entonces esto a ellos les hace confirmar un poco más la hipótesis de que el proceso que acá estaría participando es el de fagocitosis asociada al C3 o LAP, y no la autofagia canónica. En el ejemplo de abajo también lo que ven es que encuentran algunas de estas estructuras en anillo que contienen a fragmentos de mitocondria, por ejemplo, también. Y en este caso también pueden ver la colocalización con LC3. Ahora este es otro ejemplo. En este caso, esto ha sido desarrollado acá en el laboratorio en donde yo estoy realizando mi postdoc ahora, en el laboratorio de Max Gutiérrez, donde nuestro principal objetivo es las interacciones entender las interacciones huésped-patógeno en el contexto de infección con Mycobacterium tuberculosis. Y en este caso, ellos están utilizando células endoteliales, linfáticas endoteliales, LEX, que han sido crecidas, en este caso, en una placa que se llama MATEC, probablemente ya la han escuchado hablar, que pueden ver acá en el panel B. Esto simplemente es una placa que tiene una grilla con coordenadas. En este caso, por ejemplo, 2 letras, pero puede tener una letra y un número. Entonces, nosotros vamos a crecer las células en esta placa y luego vamos a poder ubicar a la célula de interés en base a estas coordenadas. Y eso después nos va a permitir saber exactamente qué célula es la que queremos adquirir en el microscopio electrónico. Entonces, en este caso, lo que se intentaba responder era si LC3 nuevamente estaría marcando una estructura de simple o doble membrana, o qué tipo de estructura era esta, porque ellos observaron de que Mycobacterium tuberculosis era capaz de crecer en estas vesículas LC3 positivas. Entonces querían entender un poco más sobre la ultraestructura de estas vesículas. Y lo que encontraron era que LC3 estaba marcando una estructura de de una complejidad bastante extensa con un montón de otras vesículas que se encontraban a su vez en esta zona. En esta diapositiva que sigue, está un poco lenta porque debe ser pesado el video que quiero mostrar a continuación. Sí. Aquí, la técnica que estaban utilizando se llama Serial Block Phase Scanning Electron Microscopy, que como el nombre lo indica, en definitiva es un escaneo en forma continua, serial, donde en este caso como está ubicado en la cámara en donde se ubica el microscopio, hay un ultramicrótomo, y este ultramicrótomo entonces es el que va a ir haciendo las secciones con una cuchilla de diamante, y cada sección se va a adquirir en el microscopio electrónico. Entonces la adquisición es relativamente rápida, en comparación con la otra técnica bastante similar que es la de FIPSEM, donde en el FIPSEM quien realiza el corte va a ser un haz de iones, en vez de ser aquí en este caso, en definitiva, el ultramicrótomo. Pero comparten similitudes, aunque también cada uno tiene sus ventajas y desventajas. En este caso se hicieron 68 cortes, y cada imagen luego se va a alinear y nos va a dar una idea en 3D de lo que está sucediendo exactamente en esta área de interés. Entonces aquí en este video van a poder observar cómo LC3 en realidad se encuentra marcando a esta vesícula, que es una vesícula principalmente de simple membrana, pero que contiene un montón de otras vesículas a su vez en su interior. Es una estructura bastante compleja, pero que permite el crecimiento de Mycobacterium tuberculosis en este tipo celular. Este es otro ejemplo que me parece genial, porque no coincide necesariamente con la forma que uno está obteniendo digamos, del patrón de fluorescencia que uno obtiene normalmente. En este caso, lo que ellos encontraron, este es un trabajo del grupo de Jennifer Lippincott-Schwartz, es que esta proteína spastin promueve los contactos entre peroxisomas y los lipid droplets. Fíjense de que en este caso se ha hecho una adquisición con microscopía de iris scan, sería casi super res, en realidad, digamos, da la resolución de super res iris scan si es adquirido correctamente, y están adquiriendo en este caso la proteína SKL, que es un marcador de peroxisomas, y en el otro panel se muestra a spastin. Si ustedes ven la forma que está marcando acá, la forma que nos genera la fluorescencia verde en este caso, no pareciera ser una estructura tubular, parece más bien una vesícula redonda. Sin embargo, cuando ellos hacen el IM y la correlación, ven de que la estructura es más bien alargada, una estructura de peroxisomas tubular que no es de lo más común de encontrar. Entonces, ellos luego realizaron FIPSEM para avanzar en todo el volumen de esta zona. Como yo les comentaba anteriormente, el FIPSEM también nos va a permitir hacer una reconstrucción 3D, luego de imágenes adquiridas de forma serial, lo que se está utilizando para hacer los cortes. Acá en este video van a poder observar la reconstrucción luego de que se alinean todas las imágenes y se hace la segmentación en 3 dimensiones. Entonces acá en rojo tenemos a los lipid droplets, y en este turquesa cian se encuentran los peroxisomas, facilitando el sitio de contacto organela-organela. Y luego en otros colores ellos hicieron la reconstrucción del resto de las organelas en esa superficie, en todo el volumen. Fíjense aquí la resolución que ellos alcanzan es de 8 nanómetros en X, en Y y en Z. O sea, es muy, muy bueno. Este es otro ejemplo en donde, en este caso, lo que se hace es microscopía correlativa de nanotubos. Fíjense la microscopía de fluorescencia. Ellos seleccionan tres áreas de este nanotubo. Los nanotubos, ustedes probablemente sepan, son muy importantes para la comunicación intercelular, pero se sabe muy poco de la ultraestructura de ellos. Y entonces, en este caso, ellos realizaron microscopía correlativa utilizando Cryo Electron Microscopy y descubrieron, por ejemplo, de que estos nanotubos contienen vesículas en su interior. Entonces, nuevamente, a partir de estas observaciones, por ejemplo, uno puede empezar a preguntarse, bueno, ¿cuál es la composición de estas vesículas? ¿Cuál es el rol de estas vesículas? Hay condiciones en donde quizás esto se genere más o se genere menos, etcétera. Son un montón de preguntas que surgen únicamente si uno tiene la posibilidad de poder observar la ultraestructura. Ahora seguramente hay interesados en, bueno, ¿qué se puede hacer si tenemos un tejido, si tenemos un órgano, y uno quiere encontrar una célula de interés en un espacio 3D, no en un espacio 2D? Bueno, en este caso ya se torna bastante más complejo, y yo estoy presentando uno de los varios ejemplos que hay. Este ejemplo también fue desarrollado por el grupo de Yannick Schwab. Lo que ellos hacen acá es una inyección de células tumorales, una inyección intracardíaca de células tumorales, y van a seguir la metástasis en cerebro. Entonces, por microscopía intravital van a poder realizar adquisición de imágenes de la vasculatura con microscopía de 2 fotones, y las células tumorales expresan GFP, entonces las podemos seguir simplemente haciendo el tracking de la fluorescencia verde. Luego, lo que se va a usar para intentar encontrar el área esta de interés donde nosotros estábamos haciendo, bueno, donde ellos estuvieron haciendo la microscopía intravital, es esta técnica que se llama NIRB, que básicamente lo que se hace es un marcado con láseres infrarrojos, cercanos al infrarrojo, de la zona de interés, que es lo que ustedes ven acá como mapa NIRB. Esta área naranja, o la que tiene el rectángulo naranja, es el área de interés, que ellos estuvieron haciendo el imaging, y a su vez hacen otras áreas, que es las que están acá con rectángulos verdes, como para tener coordenadas adicionales. Todo esto va a quedar más claro cuando les muestre un video del paso por paso, pero bueno, pueden ver cómo ya se va complejizando todo lo referido al equipamiento cuando uno quiere responder este tipo de preguntas. Y luego, esta técnica, por eso se le llama imágenes o correlaciones multimodales, va a requerir de otro paso, que es un escaneo de tomografía micro-CT, donde básicamente lo que se va a hacer es un tomograma del bloque de resina que se adquiriría normalmente en el microscopio electrónico, pero que nos va a permitir ese tomograma identificar el área de interés que nosotros previamente ya marcamos, y va a dar las coordenadas exactas en 3D para que después luego se pueda hacer el corte en la resina en las coordenadas precisas y luego finalmente sí adquirir mediante microscopía electrónica, porque si no sería muy difícil y llevaría meses quizás encontrar justo el área en todo ese bloque 3D que queremos nosotros adquirir. Acá en este video va a quedar bastante más claro. Este primero es un ejemplo de la microscopía intravital, donde se va a ver a la vasculatura en rojo y a la célula tumoral en verde. Luego ellos continúan la adquisición, en este caso 3 días después de la inyección. Van a ver que ahora el área tumoral ha crecido. Y luego entonces de haber registrado las áreas de interés, se va a llevar al microCT el bloque de resina, y con esta tomografía se va a poder hacer un registro de lo que aquí le llaman landmarks, o sea, simplemente coordenadas, que van a permitir que luego el operario pueda hacer el corte de resina en el área correcta para poder hacer la adquisición de imágenes de esa célula en particular o de esa área que rodea a esa célula en particular. Y eso es lo que muestra ahora el video siguiente. En este caso ellos utilizaron FIPSEM, o sea que hacen la adquisición de casi todo el volumen de esa área de interés que se mostraba en el video anterior. Fíjense cómo bueno, la complejidad y la cantidad de información que se puede obtener luego de, finalizada esta metodología. Otro ejemplo en donde se utilizó microCT para la localización de una célula de interés en un espacio 3D es este trabajo, donde en este caso también a su vez ellos hacen la caracterización ultraestructural de un nuevo compartimento, en este caso en células microgliales, que lo denominan gastrosoma, que es básicamente una especie de intermedio entre un fagosoma y un lisosoma. Ellos comienzan haciendo microscopía de fluorescencia y ven de que este compartimento, que básicamente ingiere cuerpos apoptóticos, comparte características de determinadas organelas de la vía endocítica, pero no es ninguno en definitivo, o sea, no pueden definirlo con los marcadores clásicos. Entonces, mediante luego la técnica de microscopía electrónica y CLEM, pueden brindar más detalles de qué es en definitiva este compartimento, y bueno, y contribuyen con el descubrimiento de este nuevo espacio subcelular que se llama gastrosoma. En este caso lo que utilizaron es zebrafish, y utilizan la técnica de microCT para encontrar a esta célula microglial de interés en el cerebro de zebrafish. Y acá van a ver la reconstrucción que luego de la tomografía serial y la segmentación en 3D, que es genial, la verdad. Fíjense cómo pueden tener una resolución espectacular en los 3 ejes, y cómo esto también permite ubicar y entender los distintos contactos que ese compartimento tiene, que sería imposible de entenderlo con microscopía en 2 dimensiones. Y bueno, y con ese ejemplo básicamente voy terminando. Quiero cerrar con con este último trabajo que fue publicado en enero de este año. No lo voy a desarrollar.
Claudio,
¿Sí?
en 2 minutos más o menos tendrías que estar cerrando. Te aviso.
Sí, esta es la última diapositiva. Este trabajo lo dejo para, si les ha despertado la curiosidad suficiente, porque básicamente merece una charla entera. Es increíble, esto es seguramente un gran paso en lo que es la biología celular, porque en todos los ejemplos que hemos nosotros, que he estado desarrollando anteriormente, tenemos una célula o un área de interés particular. Bueno, en este caso han hecho claim de una célula completa, de células completas. Entonces, imagínense directamente poder hacer correlación de toda la célula en 3D. Es realmente increíble y les recomiendo la lectura de este trabajo, pero a mí me gusta mucho el mensaje que deja porque es similar a las preguntas que empezamos discutiendo en una de las primeras diapositivas, y para mí lo que más curiosidad me despertó cuando me encontré con esta metodología, que es básicamente, dice, todo lo que nosotros estudiamos, básicamente todo en donde nos hemos enfocado, nos ha revelado resultados inesperados. Y eso yo creo que es genial porque permite el desarrollo de nuevas áreas de investigación, y ellos, bueno, comienzan a enumerar ejemplos de todos estos resultados inesperados que serían imposibles de encontrarlos de otra forma. Para los que luego quieran seguir con otra literatura, quedan acá de referencia algunos trabajos que yo no mencioné, pero que tienen explicaciones muy didácticas, inclusive con videos, como el primero de los trabajos que acá está numerado. Así que ahora quedo abierto a las preguntas. Muchas gracias. Espero no haber ido muy rápido. Y bueno, quedo esperando las preguntas. Muchas gracias.
Bueno, muchas gracias, Claudio. Muy linda tu presentación. La verdad que son metodologías muy complejas. Acá estamos esperando, hay un par de preguntas. Mientras tanto, yo te voy consultando para que comentes un poco. Una vez que adquirís las muestras, pensando en CLEM, básicamente lo que vos hiciste en EMBL, una vez que vos adquirís y obtenés los datos, el procesamiento, si requiere de softwares especiales y si requiere de una potencia de la computadora grande, o se puede hacer con PCs convencionales, porque me imagino que el tamaño de los archivos debe ser bastante grandes.
Sí, ese es uno de los aspectos importantes a tener en cuenta. Sí, esto va a requerir de computadoras con bastante memoria, que es lo que por lo general le llaman de workstations. Los software, por suerte, son open, o sea que no hace falta suscripción, y la comunidad es súper amigable, y ellos están actualizando continuamente, y si uno tiene dudas puede preguntar, hay foros, o sea que en cuanto a los software, por supuesto hay que aprender a utilizarlos, pero por lo menos los principales son gratuitos, y luego sí haría falta una computadora muy buena para el procesamiento de tomogramas. Cuando uno quiere procesar tomogramas, va a necesitar de bastante memoria. Sí.
Bien, gracias. Acá hay algunas preguntas más. ¿Es posible hacer una reconstrucción 3D aún sin tener un sistema de adquisición automática?
Sí. Por supuesto, es mejor si uno tiene la adquisición automática, pero en realidad la adquisición automática es para adquirir más de un evento y uno deja el microscopio andando, como se dice, digamos. Pero sí, uno puede adquirir 3D una sola área de interés. Eso es posible, y digamos, la forma más básica de hacer eso podría ser si uno hace el corte seriado manual, inclusive no hace falta irse al corte automático. Eso va después, obviamente, depender del tipo de microscopio electrónico que uno tenga, cuál va a ser el espesor de esa sección y cuántas vamos a querer hacer, porque luego obviamente hay que alinearlas a esas imágenes.
Otra pregunta muy simple, pero que muy realista. Dice si es posible hacer CLEM en Argentina.
Sí, es posible. Yo ahora, digamos, lamentablemente no estoy en contacto con todos los grupos haciendo CLEM en Argentina, pero como mencioné al comienzo, el grupo de la doctora Colombo en el IEM en Mendoza es uno de los pioneros, así que sería uno de los primeros a los que yo contactaría, y ellos están haciendo CLEM. De hecho, cuando nosotros hicimos el curso, hicimos una práctica digamos, es una metodología que está disponible. Luego también, antes de venir a realizar acá el postdoc, yo estuve en contacto con la gente del Centro de Microscopía Electrónica de la Facultad de Ciencias Médicas, ahí en la UNC, y ellos también estaban intentando ponerlo en funcionamiento. Básicamente, si hay un microscopio electrónico, yo sé, en Tucumán también sé que estaban intentando hacerlo. Básicamente, si uno tiene el microscopio electrónico, es posible. La forma más simple de hacerlo sería con estas placas mate que yo mencionaba, que tienen una grilla con la que uno se va a poder orientar y elegir la célula que luego va a querer adquirir. Luego uno puede hacer la fijación química, luego de tener la adquisición por imágenes, si empezamos con live cell, o se puede fijar y luego adquirir la imagen, y luego a la plaquita esa se la va a procesar para que finalmente termine en un bloque de resina, y luego por último la adquisición por microscopía electrónica. O sea que es algo posible y que tiene distintos niveles de complejidad. Yo creo que al menos en este nivel de complejidad sería algo que se puede poner a punto en lugares donde haya un microscopio electrónico.
Por último, no tenemos mucho tiempo. En un minuto, si podés contestar, si en células que no pueden ser transfectadas, podés hacer CLEM con marcación con anticuerpos sin que se transfecte la célula.
Se puede. Bueno, ahí ya hay que empezar hay que tener otros parámetros en consideración, pero sí, sí es posible. El tema es la compatibilidad de si es necesaria o no permeabilización. Si no hace falta permeabilización, es casi sí la respuesta. Si hace falta permeabilización, hay que poner a punto un poco, pero también se pueden utilizar, por ejemplo, sondas que se unan no sé, por ejemplo, a mitocondria o a peroxisomas o lo que sea, y luego uno fija. En ese caso no fue necesario el anticuerpo. Obviamente, si hace falta seguir la proteína de interés y no se puede sobreexpresar, va a ser necesaria la marcación.
Bueno, muchísimas gracias, Claudio. Lamentablemente no disponemos más tiempo, podremos seguir charlando más, pero tenemos que seguir a la próxima. Muchísimas gracias por tu charla y bueno, seguimos en contacto.
Gracias, Pablo, y saludo para todos ustedes. Nos vemos.
Gracias. Well, we continue with the next speaker. Is Dr. Javier Hagenstein. Is a CSO at Cryo Capsule company in France and Germany. Dr. Hagenstein in 2007 obtained a master's degree in cell biology and Physiology and Pathology at the University de l'Institut in Paris. In 2011, obtained a PhD in cell biology and biophysics at Heidelberg in Germany. Then has a postdoc experience in the Institut Curie in Paris. He established CLEM at the Institute. It's since 2012, he's a founder of the company Cryo Capsule. It's a company for developments and implementation in electron microscopy focused in CLEM. So he developed new technologies called Cryo Capsule to be applied in microscopy studies. He studied melanosome biogenesis and contributed to numerous publications with high quality and cutting edge technology related to microscopy, where I was applied especially in CLEM in numerous prestigious papers in a number of very high impact journals. Dr. Heiligenstein has the experience that I want to notice that of the development and implementation of technologies related to CLEM. So it's, I think, a very important talk for all the people to learn, to ask questions, because we have, I think it's a very good chance and opportunity for the students for learn from him. So his talk is about technological development to achieve insightful and efficient correlative light and electron microscopy studies. Please, Dr. Hagenstein.
Thank you very much. Thanks everyone for inviting me today, and for those of you that are listening. I will start sharing my screen now so that you can see my presentation. So I hope now you're seeing all my work. So as Pablo just said, my focus is working on technological development. And when I started my PhD, the main problem I faced was that the technologies I needed to answer my questions were not existing. So I decided to create technologies for my PhD. And this was the Cryo Capsule, and I will not go too much into the details of that. But then this is where I got a postdoc offer from Graca Raposo that presented to you just before. And she offered me to install my technology into the Institut Curie to develop correlative microscopies there. And by working here, I also realized that some other technologies were limiting. So I started to focus myself on technology developments rather than biology, although I have a training as a biologist. And so that's what I'm going to present to you today in my presentation. So one concept of correlative light and electron microscopy, and by one concept, I mean really it's depending on your scientific question, and you've seen a very broad example by Claudio Bussi just before, is in my case, looking at the sample alive by fluorescence light microscopy, which is dynamic, specific, and has a resolution of, let's say, 300 nanometers, not talking about the latest developments in super resolution light microscopy. And then things are moving in the cell biology field, and in light microscopy, we see things moving. But then we want is to be able to see that in electron microscopy. And ideally, electron microscopy will give you a fixed information, a contextual information. It could be specific, it depends on the technique you're using. CLEM is one of the examples, and we've heard about that from Claudio before. And the resolution is, let's say, 5 nanometers in plastic EM, and I will not go into the technical details of that. But then ideally, what you want is to overlay the two images to ideally understand what happened before, which is not always possible. So, but to achieve that, you have technological steps, and we've heard about that, but there are key steps in correlative light and electron microscopy. One is you need to do light microscopy. That's in the title. Then you need to immobilize the sample, and we've heard about that before. You have two ways of immobilizing the sample. The first one is to do chemical fixation. You add aldehyde fixatives to your sample that will progressively being cross-linked and stop living. Or you can vitrify your sample. So basically you freeze it, you cool it down very quickly, and it basically the water molecule stops agitating and your sample is immobilized. There are different ways of doing that. We'll talk about that later. Then you have to make the sample EM ready. So basically the electron microscope works in a high vacuum. So basically if you put a biological sample in a high vacuum without protecting it from this vacuum, it will simply dry out. The water will sublimate and your sample will just flatten and dry out. Then you need to do some electron microscopy per se, because again, we're in correlative light and electron microscopy. And finally, you need to register the images, or at least understand where your fluorescent signal is going, knowing that you have a difference in scale, which is significant in some cases. So all these steps are important, but what you need to know is that basically here, this light microscopy could happen before the immobilization or after. And that's what we've seen before in the previous talk with the live cell imaging and fixatives, or in the cryo light microscopy where we did the fluorescence after the freezing. Or you can also have the fluorescence after the sectioning, which is preparing the sample and making it EM ready, which is also something you've heard from Claudio before. So as you can see already, the light microscopy that I draw here can happen here, here, and most likely not after the electron microscopy that tends to kill the fluorescence. Again, I will not go into some technical considerations here. So my aim, my main focus during my PhD was to do live CLEM, and this is what I've been focusing on since. So the idea is to go towards live HPF CLEM, because HPF gives you the best ultrastructure preservation possible. So what you want to do is to image a sample alive and then load it into an assembly that will make the sample resistant to the high pressure freezing. So high pressure freezing, just to explain you the technology, requires to raise the pressure to 2000 bars, and while doing such thing, cooling down the temperature to minus 140 something degrees. By doing such, the water becomes more viscous and will not solidify, will not crystallize upon cooling, and you will achieve vitrification of the sample. So the problem is you need to blast 2000 bars of liquid nitrogen on your sample, and by doing such, if you don't protect it, you will just blow it away. So this is the reason why you put your sample in between two aluminum planchet or carriers, and then you load all of this into an HPM handle and then freeze and blast the 2000 bars. But going from this step to this step here, and then finally load into the HPM can take some time. Usually it takes more than a minute. And in cell biology, especially where I was doing my postdoc together with Graca Raposo, things are moving extremely fast. And so in 1 minute, your sample could be gone away and you won't find it back again. So what we wanted was to be able to do live cell imaging and vitrify the sample as quickly as possible. So what we thought was, okay, to do all this small assembly you see here inside the handle of the HPM just before HPF. But then the manual transfer takes some time and is quite variable from one user to the other. So it's not really optimal. So what we wanted was to have the sample here loaded at the end on an automated arm, just standing right above the microscope lens. So here, this is the microscope lens, and here, this is the cryo capsule that Pablo introduced to me before. And you can find some information about me on the bibliography. It's in the traffic paper. So what we want is then to be able to have the sample holder, which is standing just right above the microscope lens of the microscope, and then at a given time, transfer the sample into the HPF chamber before storing it into liquid nitrogen. So technically what you would see is the sample is observed, and then you just say, okay, I want to freeze it now. You click a button, the sample is retracted, rotates progressively, enters the HPF chamber, and then HPF is done. Once this is done, we release the sample and we store it into liquid nitrogen. So the whole process from this live cell imaging to the high pressure freezing nowadays with our machine is 1.2 seconds. So basically, this is something which has gone extremely fast, from 1 minute to less than 2 seconds. and which is reproducible because it's a machine that does the job and no longer a human. So this is something quite important, because then from one experiment to the other, we're pretty sure that it will look pretty much the same, and the duration of the gap between the last image and the freezing is registered, so you know exactly what happened and how long it took. So just to give an overview of the system, basically we have the cell heterogeneous population of cells which are expressing or not a mutation, that's up to you. We load them into the Cryo Capsule, which is a small petri dish, very advanced small petri dish, which is handled by this clamp standing right above a light microscope. And we'll come back to that point. So you just do live imaging, and at a given time, you retract the sample, freeze, and then drop it down into liquid nitrogen. And so this automated arm has been placed inside a new generation of high pressure freezer that can handle more complex parameters of HPF. So as I mentioned, the fast transfer happens in 26 seconds to be exact, in the fastest configuration. And so the sample is just right above the microscope lens, and at a given time, you just click the button and the sample will be retracted and frozen. So high pressure freezing happens like that. So when we say we blast 2000 bars of liquid nitrogen on the sample, you've seen it, it's literally blasting 2000 bars of liquid nitrogen. So we need to protect the sample. And that's one of the key problems in HPF, is to avoid blasting the sample and make sure that you vitrify it. Okay, so this system has been completely integrated together with a Zeiss microscope, as you can see here. Here we have the microscope, which is an AeriScan system, which is connected to our machine. So basically we can do live cell imaging in super resolution conditions, which is a critical question. You want to see your sample and you want to see critical event before you freeze it. And then we can transfer and freeze the system. So we've seen that now we have a machine, which is a commercial system that the company that I'm representing is selling, which can be connected to a live cell microscope. So you can do live imaging of your sample, and then at some point you just vitrify the specimen. But then we've seen in the previous presentation that some people have been developing some cryo imaging, especially Anna Sartori, who's now at the Pasteur Institute. And one of the question, or even what we heard from Harald S. and Eric Betzik and the cryo clamp, the idea is to be able to live cell to cryo image your sample in here. The advantage of cryo imaging is that you will not have any photo bleaching, which is essential because you can do super resolution because of no bleaching. And you can take as much time as you wish before you start to do your electron microscopy. So there's a critical step here in cryoimaging. And then we have the EM sample preparation. So the cryofracture, cryofib lamella, freeze substitution, whichever technique you want to use. And this is very wide open. There are as many EM sample techniques as you have like fluorescence imaging techniques. It's very broad nowadays. It's no longer just single technologies. And then the EM technology per se, where you can do FIPSEM, cryofipsem, TEM, scanning block face imaging, you name it. There are so many ways. So the idea is, where do you want to do your next step of imaging? So the first one is easy. Then you do high pressure freezing, and then you can do cryoimaging. And you could do some imaging here. Again, that's what I said in the introduction before doing the EM analysis. So let's focus on the Lincoln stage or any cryo stages. To date, those cryo stages are commercially available from Leica or Lincoln. And as you can see here, they require to use upright microscopes. But if you remember in the previous presentation, in my presentation, I presented something with an inverted microscope, meaning that you would need to have two microscopes to do your full cryoclam workflow, which is kind of an efficient way of spending money. So one of the idea was to use an inverting arm that would just transform an upright microscope into an inverted microscope and vice versa. So these periscope technologies are existing, and basically you would transform an upright microscope here in that example into an inverted microscope. So basically a microscope that is equipped here together with a cryo stage, you could have it sitting just next to an HPM. And then once you want to do live cell imaging, you put your inverted arm, and then you can go from live cell imaging on an inverted system. You remove this reverting arm, and then you would do cryo-like microscopy. So basically, we become way more flexible nowadays with these different technologies. And so depending on which lab you're working with, you have a preference for upright microscope or inverted microscope, but it's not only a preference, it's also where you want to focus. Here in that case, in the Francis Crick Institute, what they want is to do live cell imaging, which is the most critical step. So they want to see things live. So they want to avoid having an inverted arm that would expand the light path and eventually affect the imaging path, and therefore they want to have an upright microscope, and then they can retract it and put the inverter arm to put the stage and do cryo-like microscopy as the secondary imaging. In the Radboud University, this is the opposite. They are not so much focusing on the live cell imaging, and they want to really spend some time on the cryo imaging, and therefore they focused the architecture of the microscope into an upright microscope for the cryo stage, and they use this inverting arm for the live cell imaging. So as you can see now, we start to have something very flexible. With one microscope, you can start to do many different approaches using high pressure freezing, which is a key element, because high pressure freezing is to date the only way to have a close to native ultrastructure preservation in live cell imaging, in EM of thick samples, as opposed to plunge freezing, which is limited to 10 microns in depth. And so once you have this setup here, you can do live cell imaging, and here you have an overview of the entire carrier containing many cells live. And when you zoom in, this is done at live cell imaging, you have a 10x images where you can find your cell of interest, focus on whatever you want, and then do cryo microscopy, still using the 10x, and then zoom into the 100x. And here in that case, what you can see is that these cells are these ones that are exactly present in here. So you can see that we've been able to go from live cell imaging to cryo-like microscopy without losing focus of our sample. Another way of doing things is to do live cell imaging, and then you are not really much interested in cryo-like microscopy. This depends really much on your scientific question, but you want to do in-resin fluorescence. This is something we've seen already before. So the in-resin fluorescence is mostly using low-becquerel HM20 among the world. But one of the limits of HM20 is that when you use it, if you want to preserve the fluorescence for doing in-resin fluorescence, then the resin will start to charge in volume imaging, in volume electron microscopy, serial block phase imaging, or focus ion beam imaging. And so what we did together with the company is we decided to develop a novel generation of resin, which is called the R221, that allows you to do high, to preserve the fluorescence in plastic and to have high contrast in electron microscopy. I'll come back to that point a bit later. So we can do live cell imaging and in-resin fluorescence to go later on in the electron microscopy. Or you can do something where you just focus on the resin in fluorescence to do your quality microscopy. Here in that case, the samples were high pressure frozen, pre-substituted into the R221. And then we did the imaging directly on the block face. As you can see here, this is the light microscopy. This is the EM. This is the overlay of the two images. Then you can have a higher magnification of your structure of interest and find back your electron microscopy, your structure by electron microscopy to do your identification, structural identification. So this work is currently being written down, and we hope to publish a book chapter in the coming few weeks now, together with Paul Verkade and Thomas Müller-Reichert, as Claudio mentioned before. So one of the key problems in electron microscopy, as I mentioned before, is a trade-off when you want to do fluorescence and electron microscopy. The claim paradigm is fairly high here. When you have a good fluorescence microscopy, then you will have a very poor contrast in electron microscopy. And the more you increase the stains and fixatives, the lower is the contrasting in light microscopy. But don't get it wrong here. This is exponential in both cases. So basically, instead of being a slight slope here, it would drop here as soon as you add a bit of fixatives. And the contrast requires quite some fixative, but it will already kill the fluorescence before. So this is a balance which is problematic. And so the idea would be to be able to contract as much as possible so that we would have a contrast way earlier in the whole process, so that you would have to add very low amount of fixatives to have high contrast in EM while preserving the fluorescence. And so a few years ago, and I must acknowledge Roger Wepf for that, I was approached by Roger, and he said, well, you're developing technologies and you have a knowledge in plastic that we don't have. There was these papers back in 1986 that presented a new resin that would be interesting for doing this kind of very low contrast adding, because the resin was already containing some heavy metal. So he asked me whether I could develop or reproduce this resin. So I went to my chemist and we discussed, and the chemist told me, well, you know, it's going to be an explosive resin. So it's quite problematic to have that in the lab, especially for biologists. They're not really used to explosive material. And so what we decided was to start back from a blank page and try to find out how we could make a resin that would fit all our requirements for doing correlative microscopy. So it had to have very high contrast with only traces of heavy metal salt. It would need to allow water-free substitution, so like we do in HM20. It should preserve the fluorescence, meaning that when we have the whole thing, it will not kill the fluorescence. And so that would mean avoiding cross-linking with amine groups like the epoxy resins are doing. Ideally, it would remain liquid at minus 30 degrees Celsius, because this is where we do the free substitution that preserves the ultrastructure in the best conditions. It could polymerize under UV, because we are working at minus 130 degrees. It should be, of course, transparent to visible light after polymerization, and have enough rigidity to allow sectioning by microtomy or ion milling after polymerization. And all of this should remain electroconductive to give high contrast in the end. So we worked on this project and for a couple of years, and we came up with the R221 resin that allow us to do these kind of things. And so as you can see here, this is an example of the contrast, the gain in contrast we can have. Here in this free substitution approach, we have here the R221 resin, which has only 0.05% of uranyl elastate, traces of glutaraldehyde. I like glutaraldehyde in free sub because it gives crisp membranes, and 5% of water. I refer you to the Kukulski method for this 5% water. If you do the exact same experiment with HM20, you would notice that the cell here have much lower contrast. The ultrastructure is as beautiful in both cases, but yet here the contrast is much lower. If you look at the histogram, for the same number of counts, you would see that the histogram is much more spread here than it is here. In both cases, we have the same number of counts, so the same surface. So this illustrates the fact that the resin gives us a higher contrast than the HM20 with the exact same concentration, a very low amount of uranyl acetate. And this cocktail here has been used among many, many labs in the world, including Claudio before, because it preserves the fluorescence in the resin block. So we have already a higher contrast, but can we do block face imaging, which is a critical question. And so here what we can see is a comparison. This is the block surface, which is observed by scanning electron microscopy in serial block face imaging. That's why you see things dancing. And this was done with Lovicryl HM20, so the widely known resin. Here on the second video, this is exactly the same free sub conditions, so a bit higher amount of UA, no fixatives, 5% of water. And this is the kind of contrast you can obtain from our same sample block. So as you can see, we've gained a significant amount of contrast, but since we are using a very low amount of uranyl acetate, we can preserve the fluorescence. And so it starts to become very interesting because you can do your free substitution, and then you can start to image again your block of a resin before doing the free substitution, before doing the electron microscopy. And since during free substitution, you might have some minor alterations in the ultrastructure or some general deformation, shrinkage, growing of the structures, your live cell imaging might be slightly off from your block, your plastic block. And therefore doing fluorescence imaging in the resin will bring you the conviction that your fluorescence overlays perfectly with your electron microscopy. So if you take this and you integrate that into one single experiment, you do high pressure freezing, as I mentioned, you freeze substitute it with a very low amount of fixatives, and then you do fluorescence in 3D by confocal microscopy directly of your block. And what you can see here is a C. elegans that has been high pressure frozen and freeze substituted with the R221 resin, so this resin, for preserving the fluorescence. And you can see that the fluorescence remains very bright in the block, and still we can do some highly contrasted electron microscopy. ECCLAM that you can see here is the software we've been using for correlating the images, and this is a software I developed a few years ago together with Perrine Paul Gilotto, and together we made this software for easy correlative light and electron microscopy. I'll let you read about that because it's highly documented, and I think you can drop me a mail anytime, or Perrine, if you have any questions on how to use this software. And so when you have this, what you can do is you can, in 3D, start to blend the data. And then this is the kind of images you could get, where the fluorescence is perfectly overlapping with the electron microscopy. And so you can start to do precise localization of your fluorescence signal inside your electron microscopy block at high contrast. So this is really what we were aiming for, is to develop the complete workflow. So you can do live cell imaging, high pressure freeze, do cryolight microscopy if you wish, freeze substitute, light microscopy on the block again, and then do electron microscopy and have all of this with high contrast and high quality without any compromise. And so with this, I'd like to end up my presentation. I think I presented most of what we developed. I did not go too much into the details of the ECCLAM, but I guess I'm reaching out to the end of my time. I want to thank a lot of people in this work. Of course, Graca Raposo and all her team that I worked with for 6 years at the Institut Curie, and with whom I developed a lot of these technologies. And Jean Salamero, who was the head of the imaging facility and gave us significant support on all these technological developments. Roger Gadfa mentioned him during the presentation, but it was one of his ideas to develop the R221. And because he pushed me and I developed this resin. Our academic partners at the moment, Radboud University and the Craig Institute, with whom we're developing a lot of these approaches, and you've seen some of these results. The people from Zeiss that have been helping us in assembling the whole system and having an advanced microscope next to our HPF, and former people from Zeiss. We've worked also a lot with Nikon in the past. And obviously, I want to thank all the people in my company, including my dad, Xavier Heiligenstein here, who's the head of the company and who's been turning my ideas into realities. Frédéric Hérault, who's programming these technologies. Martin Bell, who's leading the company and taking care of all the business. Laurent Manet, who's the engineer that assembles the system and supports and works together with my dad to make my ideas real again. And Stéphanie Carrion, former member of the company. And obviously, all of this work couldn't be done on our own, so we've been supported financially by private equity funds and by French National Bank. And with this, I'd like to thank you, and I'm happy to take any questions.
Thank you very much, Javier, for this presentation, the beautiful images and technologies. We are waiting for questions that come from the audience, but waiting for that, I have a general question because I see that you are a real developer of new technologies. And I was thinking that these technologies allows us to see more details that we never saw before. So many things that we never imagined. So details about our cells. So, but many of the data are images, but also you get some measurements and generate data. So on the other hand, in another fields of the science, there's a huge increase in the analysis of multiparametric big data analysis with algorithms. So you have some kind of interactions between this field of the science that go to the detail with the other one that goes to multitask and multi-data analysis?
So, of course, this is a critical question, and I must say that most of the people which are in my situation, so technically I'm the guy who makes people talk together. I mean, a biologist comes up with a question and I try to come up with a technological answer. But obviously, as you just mentioned, the data set which is generated, especially in correlative, in volume CLAM, is so much that we cannot humanly analyze it, because it would take just an entire life just to segment out all of the data. So more and more of us are working very closely with image analysis. And Perrine Polgilotto, that I mentioned before, she's here in the presentation, who wrote ECCLAM, is the perfect example of that. She's at the real interface between all the biologists and all the image analysts, and she's herself a very talented mathematician who's doing a lot of analysis herself. And she's creating the link. And there are some networks nowadays which are actually developing and proposing the softwares for doing the analysis. I did not mention that in my presentation because this is something we're still working on, but my company is also currently working on proposing a service so that basically you would create the data, you send it to us, we do the analysis, and we send it back to you. But this is something which is very commercial, so I didn't go into the details for now. But indeed, this is becoming something which is multiplexed, and reasonably, no one is able to handle all of this at once anymore. Technologically, doing the correlative microscopy is a specialty nowadays, and you have specialists in CLAM, which are mostly coming from EM or LM, and they just did the bridging, but this is becoming something critical. But then someone like me who's doing some technology development cannot be a talented informatician or mathematician and be a talented biologist. I mean, at some point, I mean, some people are brilliant enough, but I'm definitely not brilliant enough. So I need to have support. And that's where I say I'm at the interface. I try to find the people which are able to help others, and I make them communicate with each other. So indeed, nowadays, we need to have access to all of this massive analysis of metadata. Also, that requires more than just a basic computer.
So it's perfect. Indeed, in this sense, is planned to create a database site where people can just upload the data set that is generated by these kind of studies?
So there's already a software, a website. I'm trying to find out the name while I'm talking, but there is a website created by Perrine Polgilotto because she's a very active leader. And there is a European network now that connects all of this and try to combine all of these informations at once. So the European network is called the COST Comulis, so C-O-S-T, further away, C-O-M-U-L-I-S, and the COST Comulis has as a point to bring together all the knowledge about correlative microscopies in a very large way and all the softwares and put that all at once into one website where you can find everything. So this European thing is already accessible. If you find on Google COST Comulis, you will find it. And it will refer you to the software database, which is online, where you will have access to all the software for doing your analysis and everything, and try to guide you into finding out what you want to do. So it's now becoming something very organized, extremely well structured, and you're no longer on your own and trying to find out into the sea, fishing out one fish. There's a big fish net and people that knows how to handle it.
Perfect. There is a question from the audience that said, I have very basic question. How do you identify the position of the cells that you have imaged by fluorescence on the electron microscope? Do you grow them on grids or the equipment record the coordinates?
So it depends on the workflow you select. In my case, in the correlative HPF, I'm using the Cryo Capsule. And as I mentioned, the Cryo Capsule is a small Petri dish, but has carbon landmarks at the bottom that will tell you you're in A, B, C, D. And that will allow you to see the sample alive and know where it is. So it's, let's say, in A2. And then you freeze, and then the carbon landmarks are transferred in the plastic, so you can find your cells of interest back. This is with the workflow I'm presenting from Cryo Capsule. But if you're doing chemical fixation, which is also something extremely broadly used, you can use MatTech dishes, which are laser graved Petri dishes. And then same applies, you will have to see I'm in B2 or in K4. We've seen some example in Claudio's presentation before. But you need landmarks to find back your cells. Otherwise, nothing looks more than a cell than another cell.
It could be difficult, yeah. Okay, thank you very much, Javier, for responding to questions and all the presentation. And it was a really, really interesting topic. I think, thank you very much in the name of the organizers and myself.
Thank you very much for your invitation.
Okay.
Bye.
Thank you very much, Javier, for this very interesting presentation and for your participation in this course. We really appreciate it. And thank you, Pablo, for sharing this session. And for everybody, I will speak in Spanish now. Thank you. Para todo el resto de la les avisamos que reanudaremos la última parte de este curso a las 14 horas. Nos vemos luego. Adiós.